ሰላም ውድ ኢትዮጵያውያንና ኤርትራውያን ወገኖቼ ዛሬ ደግሞ ጁላይ 31 ዩኒቨርሲቲ ኦፍ ቶሮንቶ ዶክተር ሙሉ ገለቱ በኢትዮጵያዊት ፕሮፌሰር ሪሰርች የምትሰራ ዛሬ እዚህ ልጆችን ከ9ኛ ክፍል እስከ 12ኛ ክፍል ያሉ ኢትዮጵያዊ ኤርትራዊ ልጆችን ላቦራቶሪ ውስጥ ወስዳ ኤግዚቢሽን ታደርጋለች። ሳይንስ መማር የሚፈልጉ መግባትና ብዙ ነገርን ኤክስፕሎር ማድረግ የሚፈልጉ ልጆቻችንን እንደዚህ ኢንከሬጅ እያደረገች ሪሊ ይሄ በጣም ሁሌ ነው ማመሰግናት እኔ ለወገን ለህዝብ በጣም እማደንቃት ይሄንን ፕሮግራም ታደርግልናለች። ስለዚህ ይሄን ፕሮግራም በየአመቱ 2 ጊዜ ሆስት ታደርገዋለች። በጣም አፕሪሺየቲቭ ነው። ለምን ብላችሁ ብትሉ ልጆች አንዳንድ ጊዜ የት መግባት እንዳለባቸው ምን ማድረግ እንዳለባቸውና አክቹሊ መጥተው ዩኒቨርሲቲ ውስጥ እንደዚህ አይነቱን ነገር ሲያዩ በጣም ይደሰታሉ እና ሰፖርት እናድርጋት። ኮሚኒቲያችን ውስጥ ነገርን የሚያመጡ ዘር ከዘር ሃይማኖት ከሃይማኖት እያመጡ የሚያባሉትን ሰዎች ሳይሆን እንደዚች እህት አይነት በጣም ለወገን የሚቆረቆር ለልጆች የሚያስብ ሰው ሲገኝ ሰፖርት እናድርግ፣ እናድንቅ፣ ሴሌብሬት እናርቅ እላለሁ። በኋላ ላብ ውስጥ የምንሰራውን አሳያችኋለሁ። እኔ ደግሞ ዛሬ ሜንቶር ልጆችን አብሬያት ቮለንተር አድርጋለሁ፣ አሳያችኋለሁ ላብ ውስጥ ምን እንደሚካሄድ በኋላ። ቴንክ ዩ። ይሄ እንግዲህ ቶሮንቶ ላቡ ነው፣ ዩኒቨርሲቲ ኦፍ ቶሮንቶ ላብ። እቺ ዲንክ ሴት እኔ ሁሌ ነው የምላት። ለምንድን ነው ብዙ ጊዜ ኦፖርቹኒቲዎች የሉም ነው ኮምፕሌን የምናደርገው። እቺ ኢትዮጵያዊት ሴት ልጆቿም አድገዋል፣ ራሳቸውን ችለዋል። ይህንን ቻንስ ለልጆቻችን እንስጥ ብላ ነው ብዙ ጊዜ የምትታገለው። ይኸው በግሩፕ ሌላ ግሩፕ አለ፣ እንደገና እዚህ ደግሞ ሌላ ግሩፕ አለ። እኔ ሜንቶር ነኝ ማለት ነው። ቮለንተር አደርጋለሁ፣ ሜንቶር ሳይሆን ቮለንተር አደርጋለሁ ዛሬ እና ይሄንን ነው የምታደርገው። ይሄ ሌትስ ጎ ጋይስ ሳይሆን እንደዚህ አይነት
which group is it? Group 4. Okay, you can come here this one and then you can come.
በጣም ኢንከሬጅ አድርጓት እና ሰፖርት እናደርጋት እላለሁ ነው። እንግዲህ ልጆቹ እየተከፋፈሉ ነው። ግማሾቹ ወደ ሌላ ላብ እየሄዱ ነው፣ ግማሾቹ ወደዚህ እየመጡ ነው። እግዚአብሔር ይመስገን ዛሬ ደግሞ ብዙ ልጆች መጥተዋል፣ ደስ ይላል። አሁን እኛ ደግሞ ወደዚህኛው ቲም ልንሄድ ነው። ይሄ ቲም ደግሞ እንግዲህ ቅድም የካንሰር ሴልን አይተናል፣ አሁን ደግሞ ሌላ ልናይ ነው ማለት ነው። ቴንክ ዩ።
Just look through the eyepiece. You don't have to touch anything. You're just going to see like circular, bright, like little bright circles. Yeah, each individual circle is a cell actually. But yeah, so this is very unique cell line, I would say, because it's like the first cell line to ever allow cells to grow. And so this cell line is actually very important because IVF is possible because of these cells. Does anybody know what IVF is? In vitro fertilization? Yeah? Do you wanna give a quick spiel on what IVF is? It's like, yeah, okay. So IVF is a process that allows doctors to take an egg from a woman and take sperm from men and form a embryo outside of the body. So that's only possible because cell culture is now possible. So the cell line is really credited for even allowing IVF to exist today. Yeah. Any questions? Has everybody looked through the cells? Yeah? Come on. Don't be shy. So just look through.
Is this HeLa cell?
HeLa. Yeah.
Okay. There is a story behind HeLa cells.
Yeah.
HeLa is an acronym. This cell taken from the patient. She is a black woman, black American. She was a cervical cancer patient. When she go to the hospital, they took the sample and then to diagnose, and then they took to the lab for research, and then they found out these cells are very aggressively dividing and growing. So the first cell line research started on the human cell lines, and they took this cell line without her consent. There was no consent at that time. She don't know what is going on with her cell line, and then they all over the world use this cell line for developing vaccine, drugs, and everything. And then later time, the family found out she didn't sign a consent, and then they do legal things, and then I think they get compensated. there is a book, very interesting book. It is The Immortal Life of Henrietta Lacks. Her name is Henrietta Lacks. They took the first two spelling in her name and then the last name, and they make Hilla. That's the cell line become Hilla. It's very interesting book. If you're interested, just read that book. The Immortal Life of Henrietta Lacks. She died, but her cells are survived 50 years. It is 50 years old.
Can you imagine?
Yeah.
What science can do?
Yeah, that is the story behind. So normally cancer cells are dividing continuously without non-stopping. So because of that become disease, you know. So we are using in the lab to screen the drugs, to research different kind of things. So we are using HeLa and other cell lines also.
Okay.
Any question?
Yeah, sorry.
Sorry.
Is there like a specific type of cancer that you guys do?
That's a great question. We mostly research cancers of the blood and the brain. So blood cancers such as acute myeloid leukemia, lymphomas, cancers of the brain such as neuroblastomas, medulloblastoma as well. We do a lot of the brain cancer work in collaboration with labs at McMaster. So yeah, there's a lot of research at McMaster as well. They have labs similar to this, and yeah. But we do some other diseases like aside from cancers. Mulu has worked on Duchenne muscular dystrophy, which is a disease affecting young boys. So their muscles don't essentially develop as they're aging, and so they actually, maybe Mulu can explain this better, but I believe they actually degrade and
degenerate.
Degenerate.
Sorry. Yeah, this Duchenne muscular dystrophy is affecting boys only because boys are getting this mutated gene from mom. Mom is recessive of this gene. When boys are formed, they are taking X and Y chromosome. So if they are getting this defected X chromosome from mom, they got this disease. They diagnosed when they are 4 years old because they are not climbing the stairs, they are not walking properly, and then when they become teenagers, they are on wheelchair. They cannot move because the muscle is degenerated. It's not developed. It's very weak. And then when the life expectancy is 20, 25 years old, and then they passed. It's very devastating. In our lab, what we are doing is, in the lab, we grow the muscle cells, and then we make to regenerate the muscles using our drugs that the chemists making. So we add the drug to the muscle myoblasts. There are myoblasts. We add the drug and then wait if they are differentiating or not. So we wait a couple of days, and then if the drug is good, it's a nice one, if the cells can differentiate, we can see under microscope. So I don't have in culture these cells, but I have a picture here how we make muscles on the plate, on culture dishes. For example, these are muscle cells plated on the plate and waited for a couple of days, and then we added some drugs that are produced in our lab. This is a control that we have. So you can see the muscle fibers developed on the petri dish and looking different proteins, muscle-specific proteins that are making muscles, the proteins that are making muscles. So see the muscle fibers here, it is on petri dish. So we are looking different proteins. For example, this one, the green one is acetylated alpha tubulin, the protein, and the red one is caveolin 3. It's muscle-specific protein, and then muscle. There are another protein, muscle-specific protein is myogenin. So it is inside the muscle. When the muscle form, the cells are fusing nucleus, because one cell has one nucleus normally, yeah. But when muscle develop, they have several nucleus because muscle has energy. Yeah? Everything is done with muscles to produce energy. So it has multinucleated. So because of that, but this patient, the muscles are not developed. It's not fused. It's degenerated. So muscle become weak and it's not working. So because of that, we are trying to find a drug that is regenerate the muscles. So that is genetic diseases. It is very rare disease, but it's very devastating. Your boy if he got this one, he cannot survive more than 25 years.
So the treatment starts like at what age, or just since they were?
There is no cure for this disease for now. There is no drug. We are trying to develop a cure, you know.
Okay.
Yeah, there is management. There are several drugs to manage them. Because this later day it become heart problem, because heart are muscles.
Yes.
It become lung problem. Lung has muscles. We have everywhere muscles. So it maybe it heart attack or something happens later age. So there is no cure for this. But management, to manage the patient, there are some drugs, but not curable.
How is the percentage? Like how many?
It's very small. Yeah, it's very little percentage, but we started this project because one of our staff here in the campus, his son developed this thing. So he raised money for this research fund and donated to this lab to develop research for this disease. But most of the time we are working on cancer drug development. So we are working on leukemia, breast, and cervical, lung, different cancers. So we are working with that. Any question for me or for Simran, you can ask. So
Yeah. If it comes from the X chromosome, how come it doesn't affect women too, like in girls?
girls are not affected because they become carrier. Because they have another good X chromosome. Girls are XX, yeah? Boys are XY. If they got the damage or the bad or mutated X, and then the good Y from that, and then he will be sick. But if he gets from mom normal X, and then the Y, he will be normal. But the girl, if they get X from the mother, the mutated, and the X from the father, they are okay. Yeah, they become recessive. They pass to the other generation, you know, they become recessive. Yeah.
That's it I'm missing, guys. Thank you so much for your time, it's a precious thing to be, I don't know, I always dreamed to be born in this country because we never had this opportunity to see the cells before even you became a doctor.
Yeah.
Or in university. So thank you, Muri. I always appreciate you for
You're welcome.
giving the opportunity for these young people. And thank you for your time. It's my second time meeting her.
Yeah.
Yeah, I had experience with you about something also. We were talking.
We also
okay.
we also store our cells. We are not just growing. We grow some cells for our experiment. So it's 37 degrees centigrade, like body temperature. Yeah. If it's not body temperature, mimicking body temperature, they are not grow, you know. So we mimic body temperature. But the rest of the cells we have to freeze. So Simran will show you where we are freezing the cells. So this liquid nitrogen is 180 minus degrees centigrade. So we freeze them. You can keep them as much as you can. 20 years, 30 years, you can keep the cells. So I have several cells here. I can show them. You see, we can save like this the cells. Wow.
It's very cold. The liquid nitrogen.
Careful.
Yeah. So we can save like this. Whenever we need for experiment, we just take out one and then thaw them, and then we have media to grow the cells. So this is a biosafety cabinet. It's very sterile. Nothing there happen. So we work inside because we don't want to contaminate with the outside air. Outside is several contaminants here, bacteria, viruses, and everything is on the air. So we work on a sterile condition.
So this is how the HeLa cells, for this patient back in the 50s or 60s.
Yeah.
This is how her cells have been able to, this is how we've been able to preserve her cells for that many decades.
Just hers?
Well, hers is like being one of the first cells,
yeah.
but no, it's all kinds of patients. There's probably hundreds, maybe thousands of cell lines. Of course, identity is protected after learning from the mistakes with Henrietta. But yeah, identity is protected, but there are cells all over at research institutes, hospitals, stored like this.
But this time they just take consent, you know. You have to fill the consent.
And
so
of
the
course-
patient agree.
Yeah.
So to do research or something, to take their cells for research. So we have tons of cells in this. So to do this medical research. Yeah. Any more questions?
Does it have to be a specific kind of cell, or
It depends.
Yeah, it
like blood
depends.
cells? Or-
Yeah, we have blood
we have
cells.
blood
as
cells. I
well.
can show you blood cancer cells. I have.
Yeah, so from AML, they can be blood cells. It can be brain as like
See, this is
neurons
the
as
incubator
well,
that we're
for example.
storing the cells.
Is there any cell that can't be like a specific cell you can't use, or all of them?
I think for the most part, it's about having a patient that consents to, the cells
This
being
is,
distributed or like
Chronic myeloid
extracting
leukemia cell
the
lines.
cells, so, as
So we
long
are
as
growing.
it's safe
There are acute
to do
myeloid
so,
leukemia, chronic
to
myeloid
extract the
leukemia,
cells from the patient.
different kind of leukemia. You know leukemia. What is leukemia? They are blood cancer. So this is one of the blood cancer cells that we have in culture. These are floating cells, The solid tumors, for example, breast, cervical, and what is it? They are stuck to the surface of the plate or the flask. But this is blood. Blood is fluid, yeah. So they are moving here and there. The blood cells are
They're good.
not adherent. So you can see them. They are very tiny, circular, and you can have a look.
Diverted to community social work.
Tiny circular things, but the others are spread and stuck on the surface of the plate or the flask. Just go ahead. You can have a look. You can adjust this one for your eyes because everybody's eyes are different focus. So you just adjust to see the cells.
No, it's great.
And having
Yeah.
experience in hand.
Did you see something? What did you see? Yeah, that is the blood cancer cells.
You just memorize it, and that's
Yeah.
the question. Versus knowing this.
Yeah.
Really,
Yeah.
yeah.
Exactly.
That's just amazing.
Yeah.
That's what I encourage young generation to take the opportunity.
So it's
Yeah.
we usually working with cancer cells because they do the culture. Do you ever work with non-cancer cells?
Yeah, we
Yeah.
work with healthy cells. Generally, when you're doing like drug discovery, you do want to make sure that anything that you're making isn't damaging or toxic to healthy cells. So think about it when you're taking medication, you want it to target the area that's causing problem and not necessarily just kill cells, anything in its path. So we do culture healthy cells. They're called, there's various types. There's MRC9. Actually, I believe Wenlong is working with the MRC9 cells, so they are healthy cells that he's working with right now.
Lung like fibroblasts. Yeah.
Lung from the lung. So they're like the surface tissue of the lung. But then there's other types as well. I think HEC are healthy cells from the kidney. So you generally want to have both so that you can make sure that anything that you're making isn't harmful to the healthy cells. Good question.
Yeah, because we are screening drugs, just to find out the good drug. So this good drug doesn't kill the normal cells. So we have to have the normal cells together with the cancer cells to get the better drug, to decide which one is the best, which one is the best drug to be further researched or further do more experiment. Yeah. Any more question?
Everybody, you want to move slightly down and so you can see everything? Yeah, keep going because this will be the station you want to see. First, I guess we'll just have a little chat and see what you guys' interests are, and then we can go into this really cool experiment that we are going to do. So, hi, I'll introduce myself. My name is Bilal. I am a third-year PhD student in this lab. So I'm a grad student. I did undergrad before, and now I'm doing my PhD. And I've got a cool experiment to show you, but before that, I just want to get a read. What grade is everybody in? 9? Raise your hand. 9? 10? 11? Okay, 1 11. 12? 3 12s, okay. So you guys are looking forward to undergrad. Into undergrad? Yeah, awesome. What school? Awesome. Downtown? Okay, that'll be really fun. And you know what program you'll be picking? Oh, awesome. Okay, so this is applicable to that because you'll learn a lot of the stuff, the theory behind this experiment in first year. But you guys can look forward to that, right, in a few years. What we have here today is a pretty cool experiment, right? It deals with identifying an unknown amount of protein. So everybody has eggs for breakfast, right? Yeah, mostly. So eggs actually have this protein in them. In the white part of the egg, there's a protein called albumin. And in this tube, what I have is purified albumin. And we can extract the purified form, and we can use it to quantify for our experiments, right? So if I were to hand any one of you this tube of albumin, right, the protein from the egg, how can you figure out how much of it is in here, right? There's a certain technique we use to quantify that, right? To quantify, put a number value on that or a measurement for how much there is. And we're also going to use a couple more things. So we have albumin, an unknown solution. We have some water. We have some water here, and we have a unknown. Well, it's sort of known, but this is a copper solution. So you guys can see this is a green solution. It came as this set. So this is the set we used, right? So there's an activating solution. There's a copper solution in here. It's blue before. And here it is green once you mix them together. And this will tell us how much protein we have in the unknown tube, right? How does that work? Well, the copper will bind to the albumin in this tube, right? And change its color. So the previous group has done the experiment already. And as you can see, this is the one with water, and this is the one with albumin. You see how it changes to purple? So once the copper reacts with the protein, specifically the backbone of the protein, it is going to change color because the copper is going to be changing its charge state. So you guys have taken chemistry, you know what charge states are, +1, +2. It is going to change that copper's charge state and change the color as well. Now, you guys should get some experience doing this as well. So we'll get you to put on some gloves and try this out. So I have small gloves here. So who wants to go first? Yeah, you have to be... Okay, perfect. I'm going to take some. And here you go. And we'll get one more person out of you three. Whoever wants to volunteer, go for it. Perfect. Okay, so this color, this purple color, has a certain intensity, right? So if we have a lot of protein, it is going to be very purple. If there is less protein, it's not going to be as purple. And we can put this into a plate reader, and the plate reader will shine light onto this well and read what comes through the other side and put a number on that value, right? So if it's really intense, really purple, it is going to have a higher value, and if it's light purple, it's going to have less value. Water shouldn't have any, right? It should be just 0. And that's what you see here. This one's green, the same solution we put in. This one's really purple. And what we can do is use that value in a formula to figure out the concentration of protein in this tube, and then use this for experiments that we want to do with that protein, for example. Okay, so what you're going to be using is a pipette. So this is called a micropipette.
Laser tools into a port, and you can imagine that port is kind of like the abdomen, or like it's trying to insert the tool inside of the patient's body. And you can see that how you can manipulate the tool from using this robot. And the idea behind this project is that the robot that's doing this motion, it's very cheap and affordable compared to the robot that was demonstrated earlier by Radian, which is like a medical surgical robot, but it costs like millions of dollars. And so not a lot of researchers have access to that kind of robot. And so what we've done here is we've taken one of the surgical tools, which are much cheaper, and we've created like a toolbox, which is that blue shaft that's on the tool. And it essentially allows you to take this surgical tool and mount it to whatever robot you want. And then you can essentially try to replicate the surgical scenarios using any other robot. that's much cheaper, and you can like develop some algorithms and work on like figuring out how to manipulate tissue as easily as possible. And so it's much more accessible, and that's kind of the idea behind this project, which is to make surgical robotics a more accessible field for other researchers to get involved here and to start developing their own ideas rather than having to like purchase such a very expensive robot. And yeah, that's pretty much the idea behind this project.
So are they accessible to many hospitals or just like?
So all those things are just 3D printed, like in the blue part there, where the tool is mounted. And so that part is like very cheap. Like 3D printers are more like accessible, and so it's not actually used in hospitals. It's more for like a research environment.
Oh, okay.
So people can like work.
That was
with
my
this,
question.
Because like not everyone has like the medical robots in their lab.
I see some of you are tired. Any questions?
Yes.
From this side?
long run.
Unfortunately, this session is the last one.
Yeah.
After running through the entire campus for a while, I can understand. Like pretty much exhausted. Good note to kind of finish off.